Here we are, Module 15. You'll be happy to hear the majority of this module is going to be a review as we start to integrate all of the pathways we've examined so far. The first part of this module is going to look at, again, amino acid metabolism and specific roles of amino acid metabolism in various tissue types. From there, we're going to review the integration of our metabolic pathways in both the fed and the fasted state. And then we'll use a few examples of aberrations of metabolism that precipitate with a metabolic acidosis, hypoglycemia, or hyperglycemia as a means of applying all of this information. The chapters in your textbook, chapters 3, 36, and 42, will be helpful with this material, although we won't be strictly adhering to the flow of those chapters. So again, we'll start this module looking at the integration of protein catabolism with other metabolic pathways. And we'll really look at tissue-specific roles for various amino acids. From there, we'll review the comparison of the fed and fasted states. We'll revisit the metabolic pathways that are active under each of these conditions, the hormones that regulate each of these pathways. We'll also revisit the allosteric activators, inhibitors, and covalent modifications of the regulatory enzymes. Additionally, we'll review the preferential fuel for each of these tissue types, skeletal muscle, liver, brain, and adipose, under both the fed and fasted state. Finally, we'll use some case scenarios of diabetic ketoacidosis and medium-chain acyl dehydrogenase deficiency to illustrate integration of these pathways and to look at compensation of metabolic acidosis. This slide nicely summarizes the flux of amino acids within the blood. Amino acid levels within the blood are not tightly regulated like glucose levels are. Depending on the levels of protein synthesis and degradation, as well as dietary intake, the pool of amino acids can fluctuate greatly over the course of the day. So inputs into the amino acid pool come from dietary protein that we've seen is digested into ditripeptides and single amino acids. Amino acids can also come from endogenous protein degradation. To a small percentage, amino acids can be synthesized within the body, starting with dietary glucose, which can be aminated to synthesize an amino acid. Any amino acid that can be synthesized readily within the body is considered a non-essential amino acid. For draws out of the amino acid pool, we have protein synthesis. So anytime there's synthesis of new proteins, this will draw out of the amino acid pool. On average, there's 300 to 600 grams of protein turnover in the human body a day, so this is not a trivial turnover between protein synthesis and protein degradation. Amino acids also contribute nitrogen groups to any number of nitrogen-containing compounds. We looked at the synthesis of purines and pyrimidines, which both include nitrogen-containing ring structures. We looked at the synthesis of heme, and all of these require amino acids for synthesis. It can also be taken up by the liver and other peripheral tissues to be utilized in several different pathways. This can also happen in the fed state. In either case, the uptake of amino acids involves transamination to remove the nitrogen-containing group from the carbon skeleton of the amino acid. The nitrogen, or ammonia, can be used to generate an ammonium ion in the kidney as a means of secreting excess protons, or can be used to synthesize urea in the liver. The carbon backbone can be used for several processes. It can be used to synthesize glucose, in the case of gluconeogenic amino acids. The glucose can then be used to support blood glucose in the fasted state, or can be incorporated into glycogen. Or in the case of ketogenic amino acids, the carbon skeleton is used to synthesize fatty acids. So as we've just seen, the amino acid pool is constantly in flux. So within the body, the organ systems work together to ensure that the necessary amino acids for each specific tissue type are always available using this sort of inter-organ exchange. It's a means of ensuring that each different tissue type has the required amino acids to maintain function even during fasting conditions. What you can see here is the skeletal muscle is really the primary distribution site of amino acids. Skeletal muscle primarily catabolizes branched-chain amino acids, and in doing so, it can release either branched-chain amino acids themselves, glutamine, alpha-keto acids, alanine, or any number of other amino acids. The branched-chain amino acids are taken up by the brain. Within the brain, these are required for neurotransmitter synthesis. The brain, in turn, releases glutamine that can be used as a fuel source in the kidney, as well as providing ammonia used in buffering. The kidney, in turn, releases alanine, which can be taken up by the liver and used to synthesize glucose. You can see how these tissue types are connected in their interdependence on amino acid shuttling, and we'll look at each of these specific tissue types and their role in shuttling amino acids through transamination reactions from one tissue type to the next. Within the kidney, glutamine facilitates two primary reactions. First off, it's deaminated by glutaminase, and that provides a free ammonium which can absorb excess protons and provide a buffering system in the urine. This allows for the reabsorption of bicarbonate to a greater extent. The glutamate can be completely oxidized in the TCA cycle after it's deaminated by glutamate dehydrogenase. The free alpha-ketoglutarate can be used to synthesize glucose, pyruvate, serine, or glycine, and the primary role here is to provide necessary compounds for the kidney itself. So the glucose is primarily oxidized by the kidney. In some cases, the kidney can be a site of gluconeogenesis, such as in the case of starvation, but the body is not wholly reliant on the kidney for this process. The complete oxidation of glutamate is shown here, where glutamine is again deaminated by glutaminase. Glutamate is deaminated by glutamate dehydrogenase to generate alpha-ketoglutarate. From here, alpha-ketoglutarate can be oxidized in the TCA cycle to oxaloacetate. Oxaloacetate, once into the cytosol, can be converted to PEP by PEP carboxykinase. From here, PEP can either be used to synthesize glucose through gluconeogenic reactions, or the pyruvate can be used to synthesize alanine, which can be interconverted into serine. Pyruvate can be used to generate lactate, or the alpha-ketoglutarate can be completely oxidized. So glutamine, in the case of the kidney, provides substrate for energy as well as ammonium for buffering. The buffering is shown here, where the ammonia can absorb a proton within the renal tubular cell to generate an ammonium ion. The ammonium cannot be taken back up. It can't diffuse back through the plasma membrane as it's a charged molecule and is excreted in the urine. This is one means of biological buffering to excrete excess protons. The second site of amino acid catabolism we're going to talk about is the skeletal muscle. Within the skeletal muscle, the catabolism of branched-chain amino acid plays a primary role in this inter-organ relationship. Branched-chain aminotransferases are highly expressed in all tissues except for the liver. These enzymes transaminate branched-chain amino acids to alpha-ketoglutarate, generating the alpha-keto acid and glutamate. Glutamate facilitates what's referred to as the purine nucleotide cycle, where the glutamate is used to synthesize glutamine using glutamine synthetase, and a free ammonium ion generated by the conversion of glutamate into aspartate in the purine nucleotide cycle. It's through this process that glutamine is generated within the skeletal muscle or other tissues and can be released into circulation. This process is shown here in more detail. Our branched-chain amino acids, isoleucine, leucine, valine, and again isoleucine, can enter into the TCA cycle as either succinyl-CoA or acetyl-CoA. From here, the succinyl-CoA can either be used to synthesize alpha-ketoglutarate, which can be transaminated to glutamate, and can be converted to glutamine using glutamine synthetase, or can be used to synthesize malate, which can be converted to pyruvate, and transaminated using the alanine aminotransferase to generate alanine. It's through these pathways that we can generate two non-toxic carriers of ammonia within the body. Those are alanine and glutamine. This interrelationship is referred to as the alanine-glucose cycle shown here, where the skeletal muscle releases alanine through branched-chain amino acid catabolism. The alanine shuttled to the liver is transaminated to pyruvate, the enzyme here being alanine aminotransferase, and the pyruvate can be used in the synthesis of glucose through gluconeogenesis. The catabolism of amino acids in the intestinal epithelial is the same regardless of whether it's from the diet in the postprandial state or from the bloodstream in the postabsorptive state. In most cases, the amino acids are oxidized or converted until we've generated pyruvate or alanine, which can be taken back up by the blood as alanine or lactate and sent into circulation. Similar to the kidney, glutamine is deaminated by glutaminase to generate glutamate, which can undergo a dehydrogenation reaction, which can undergo a reaction with glutamate dehydrogenase to free an ammonium ion and generate alpha-ketoglutarate, which can be used as a substrate within the TCA cycle. And finally, shuttling of amino acids into the brain is a little more regulated as the astroglial cells and the neurons both synthesize glutamine as it's required for generation of many neurotransmitters. The substrates for the synthesis of glutamine primarily come from branched-chain amino acids, which are transported across the blood-brain barrier, are deaminated by branched-chain aminotransferases to generate glutamate. From here, a glutamate can be used to synthesize a glutamine using glutamine synthetase. And now let's look at nitrogen balance. So we've seen the flux of amino acids from the amino acid pool in the blood into various peripheral tissues where they're utilized for various reactions, transaminated, or used as substrates for gluconeogenesis or other synthetic pathways. So what happens in the case of too much or too little protein within the body? If synthesis of protein exceeds degradation, then you are in what's considered a positive nitrogen balance, meaning you're retaining more amino acids than you're excreting. This occurs in the case of growth, such as in children, pregnancy, or repair, where the rate of synthesis of proteins, again, exceeds the rate of degradation. Conversely, if degradation is greater than the amount of protein intake, then a negative nitrogen balance occurs. And this happens in the case of extreme or severe burns and injury, diseases such as sepsis, and starvation when protein catabolism far exceeds dietary protein intake, and an individual is in what's referred to as a negative nitrogen balance. Otherwise, nitrogen balance is considered neutral. Here we're looking at the disease progressions of two different infection types. You can see in both cases there is an initial elevation in temperature in the host. This would suggest there is a significant immune response. What you can see is over the time of an elevated temperature, the nitrogen balance becomes negative in both cases. And over the course of disease progression, the cumulative nitrogen balance is also negative. So what is it about the immune process that precipitates a negative nitrogen balance in the course of different types of infections? That's summarized here. Eliciting of the immune response through bacterial products or any number of infective processes activates the immune cascade starting at the level of the macrophage. Macrophages secrete any number of cytokines and chemokines. Here is shown TNF-alpha and interleukin-1 and interleukin-6. First off, the synthesis of these polypeptides requires amino acids. So activation of the immune system itself requires amino acids. Likewise, these cytokines will elicit the synthesis of glucocorticoids. Glucocorticoids initiate protein breakdown or catabolism from the skeletal muscle, such that amino acids are then shuttled to the liver to be used in gluconeogenesis. This also contributes to the negative nitrogen balance. So during this acute phase of the disease process, you have increased amino acid uptake of the liver, increased protein synthesis by both the liver and through the immune system in conjunction with protein catabolism. These catabolic processes are not normally a huge issue for well-fed individuals, but in the case of malnourished individuals and infections such as this, where you have decreased protein intake and an extreme immune response, can precipitate a negative nitrogen balance that can be potentially life-threatening. So let's look at this question to illustrate positive and negative nitrogen balance. A 4-year-old child after a meal will display which of the following metabolic patterns? Well, in this case, we have a young child. So we're looking at the case of growth and development. This would suggest a positive nitrogen balance, limiting our choices to A, C, and E. The question also says immediately after a meal. Let's assume this is an adequate meal with an adequate distribution of protein, carbohydrates, and fat. So our ratio of insulin to glucagon will be high. Under these conditions, neither gluconeogenesis or fatty acid oxidation should be occurring. And now, if I look at this table, I realize there is not a choice here that suits. This individual would have a positive nitrogen balance and would also have low levels of gluconeogenesis and low levels of fatty acid oxidation. So again, this table does not actually have the appropriate answer, which is taken from your book from another question. But in the case of a 4-year-old child after a meal, you would expect a positive nitrogen balance, no or little gluconeogenesis to be occurring, and no or little fatty acid oxidation. You should look at the end of your textbook, as there's several questions that focus on this table and relating flux through each of these pathways as it relates to nitrogen balance.